All right, welcome back everybody for our next section. I hope you enjoyed the poster session. Just a reminder to vote and help our students out with just a little extra cash. You'll also be able to vote on the night talks, which is what we're going to be starting here soon. If you don't have it, the ballots are down here. You're welcome to fill those out. I also just wanted to point out the fact that the posters will be up around Hatfield for the next year. And so if you didn't get a chance to look at them, they'll stay up out there in the auditorium in the atrium today and tomorrow, and then they'll be placed around Hatfield. So you're welcome to see them there. If you are a student who had a poster last year, your posters have been pulled down to make room for the new ones, and they're over here. So you can pick them up after the night talks. So just letting folks know what's going on there. Um, I'm not sure everyone's seen a night talk, but these go really fast. Our students have been given only 5 minutes. The slides are going to advance on their own. And so it'll be fun to see how we navigate this together. We're excited to have last year or the year before's winners present Ignite Talks today. And we have 10 different graduate students presenting from OSU's comparative health sciences, from fisheries and wildlife, from CEOS, from IB, marine resource management, and microbiology. So we have lots of variety of folks that'll be speaking today. And so I'll basically just give a little intro to our students, and then they'll come on up and we'll run through them. We'll have time for one single question after the talk. So if you have a burning question and you would like to ask it, when the presenter is done, just raise your hand. Again, Mariana or I will come around with a mic and we can get your questions answered that way. But again, if you have questions for our students while they're presenting, another way you can get to them is to talk to them at dinner tonight or see them around campus. So this will be fun. Is everybody ready for this? Yeah. Okay. So our first presenter is Kira Bankhead. She is working on her master's with fisheries and wildlife and Dr. Cantor. She received a 2023 Cribbins Award. And the Cribbins Award is a memorial fund for Joan Cribbins, supporting graduate and undergraduate students pursuing marine science and related fields at OSU. So I'm going to hand this off to Kara.
Okay, I'm ready. Hello, everybody. My name is Kira Bankhead. I'm super excited to be here today to show you what I've been doing over the past year on my thesis research, which I've been investigating how bottlenose dolphins in Sarasota Bay, Florida, navigate some of the challenges presented in the Anthropocene and how these have been impacting their social cohesion as well as the organization of their communities. So one example of an anthropogenic effect in Florida is red tide events, which have been increasing in frequency. For dolphins, this means the depletion of many of their primary prey species, which has led to them resorting to different food sources, such as anthropogenic or food sources provided by humans. These include begging, scavenging, depredating, as well as fixed gear foraging. Unfortunately, these have a lot of consequences for the dolphins, one of which is decreased reproductive success. And this, although we know a lot of the physical consequences of these behaviors, it's a lot less known what the social consequences of the behaviors are. And these are really important since dolphins rely on each other for their reproduction and survival. So this has led to my research questions in which I wanted to know if dolphins assort their groups based on human-centric foraging tactics, and if this kind of assortment increases during red tide. So I worked with Sarasota Dolphin Research Program, who provided me with 18 years of photo identification data, where there was a red tide event in the mid-2000s. So I subsetted my data into 3 different time periods before, during, and after this event. Then I used the photo identification data to figure out which dolphins are hanging out in groups with each other, then calculate the proportion of time any two dolphins spent associated with one another. And then with this association data, I could create networks where the dots represent individuals and the lines connecting them represent the proportion of time they spent together. Then to answer my research question, I could use that network, relate it to the similarity in their human-centric foraging behavior, and see if the network was assorted by this human-centric foraging behavior. So here are my results. It's a lot of Bayesian stuff, so I'm just gonna summarize it. I found that before the harmful algal bloom event, dolphins did seem to assort themselves based on this behavior, but this kind of assortment greatly increased during the harmful algal bloom event. And I did find that the model showed after the harmful algal bloom event that the assortment went back to the before kind of levels. So what does this tell us? First, I did find that human-centric behavior indicated how dolphins associated with one another, and that this increased during red tide. And this has a lot of consequences for the dolphins. As you can imagine, if they're assorting themselves based on this behavior, then because it's maladaptive, it can wipe out an entire demographic. So thank you so much for coming to this. I had to speed talk so fast, so if you have any questions, please let me know. Thank you.
I told you these were going to go fast. Nice job. You can breathe now.
Yeah.
Does anybody have a burning question for Kira? Look at that. They're going to let you off the hook.
Yay.
All right. We're going to go to our next presenter.
Thank you.
And that is Chris Bauer, who is working on a PhD in IB with Dr. Barreto. He received a 2022 Hill Award. And I have to flip the pages because I don't have these memorized yet. The Lewis Maud Hill Coastal Marine Studies Award is a family foundation award that supports the mission of the Coastal Oregon Marine Experimental Station of applied, interdisciplinary, and collaborative research in fisheries, aquaculture, seafood, and coastal resource that impact Oregon. And so I'm going to hand it off to Chris. Yeah, so there'll be a little delay, but we'll do this together. You ready?
Sure.
Okay.
Can everybody hear me? Yeah? Cool. No? Yes? Let's reset it. Hi, everyone. Thank you all so much for being here. I'm really excited to share about some of the research I've been able to do under the Lewis and Maud Hill Award. But before I begin, I want to talk about our coastline and how it's being impacted by climate change. The Oregon coast is one of the most biologically productive coastlines on the planet, holding a multi-billion-dollar value through industries such as fishing and tourism. And what supports these industries is an abundance of healthy and diverse marine ecosystems found from our rocky shore to thousands of feet below the surface. What facilitates this biological abundance is an oceanographic process known as upwelling, in which seasonal winds drive coastal waters offshore to be replaced by cold, nutrient-rich waters from the deep. On our coastline, this happens every year between the months of May through August, and this added nutrients essentially fertilizes our water column and leads to huge increases in the abundance of plankton, which serve to feed virtually every species on our coast, including many of the species that feed us. But the flip side of upwelling is that while this water is nutrient-rich, it is also oxygen-poor. And when it's deposited onto the coastal shelf, where a majority of these marine ecosystems lie, a phenomenon known as hypoxia can occur. Hypoxia simply refers to water that is oxygen deficient and therefore stressful to a majority of the marine life within it. And while it used to only happen in the farthest and deepest reaches of our coastal shelf, climate change has driven it to become a fixture of the upwelling season, happening at unprecedented spatial scale and intensities every year since the early 2000s. This has led to multiple mass mortalities in our fisheries, and as a result, it's through monitoring these fisheries that we've been able to infer ecosystem health during and after hypoxia. But our ecosystems are made of much more than just our fishery species, which led me to ask this question: what about the plankton? I wanted to know more about how hypoxia was impacting this important group. And so I decided to look at a species of copepod called Calanus marshalli, which is a dominant plankton species throughout the Northeast Pacific and an important prey item for multiple fishery and marine mammal species such as fin and right whales. We know almost nothing about how this species is impacted by low oxygen conditions. And with that in mind, I wanted to ask these two questions. One, how does Calanus marshalli's tolerance to hypoxia fluctuate throughout the upwelling season? And two, does the extent of breathable space in the water column for marshalli change during the upwelling season? In order to answer these questions, I was lucky enough to go out with the Peterson lab here at Hatfield on their monthly plankton surveys off of Newport. I went through on surveys May through August, where I collected environmental conditions like temperature and oxygen, as well as a live plankton sample that I brought back to the lab for experiments. Once in the lab, I sampled marshalli out and actually performed what are called respirometry experiments on marshalli, where I took individual copepods, placed them into these tiny wells filled with seawater, and monitored oxygen consumption over time, which you can see on the right. Oxygen on the vertical axis decreases until it eventually plateaus. And the point at which it plateaus is what we call P-crit. P-crit symbolizes the hypoxia tolerance of the individual. And when we take this for multiple individuals every month, we can examine how hypoxia tolerance fluctuates over time. Here, I'm showing you a box plot of P-crit values taken at May through August at 8 degrees Celsius. And what you can see is that these values steadily increase throughout the summer, which means that tolerance to hypoxia is actually decreasing from the beginning to the end of the summer. At 12 degrees Celsius, we see a bit more variation each month, but overall the pattern stays the same with an increase in P-crit, which leads to a decrease in tolerance to hypoxia from the beginning to the end of the upwelling season. Finally, at 15 degrees, we see similar effects to the other two treatments, which means that while marshalli might be able to mount a robust response to hypoxia at the beginning of the season, by the end, this response is largely diminished, which brings us to our second question about breathable space. We can actually answer this question by combining our P-crit data and then our environmental data to create heat maps of where marshalli are most likely to be in the water column based on environmental conditions, which I'll show you here. So the top of the plot is the surface. This is a sort of cutout of the water column off of Newport. Black is sea floor. Pinks and yellows represent well-oxygenated space, and purples represent less oxygenated space that marshalli is not likely to be. So in May, you can see that pretty much the entire water column is very well oxygenated, but in June, this deeper water starts to get less and less oxygenated through upwelling, which continues in July, and by August, a majority of the water column is actually so unoxygenated that it's likely driving marshalli to the surface, which indirectly could impact our coastal industries by changing the behavior of their predators, like the marine mammals and fisheries that we depend upon. That's all the time I have. I quickly want to say thank you so much to the Lewis and Maud Hill Family Foundation for providing this generous funding, as well as the Peterson Lab, Alaka crew, and my amazing lab members for helping me get this research done last summer. Thank you very much.
Don't go anywhere yet. Hang on a second. All right. Does anyone have a question for Chris?
Question?
All right.
Yeah.
Go for it.
So the biggest difference that I saw was at the 8 degrees and between the early months and August. And I'm just wondering if the Calanus marshalli were bigger in August and possibly needed more oxygen, and maybe that was related to your P-crit value.
That's a good question. So the answer is no. They actually were smaller in May. We had a mixture of different life stages. We primarily focused on adults, but we also focused on Copepodite 5 life stage, which is just before the adult stage. So it gets a little bit more complicated, but I'm happy to talk about that with you afterwards.
Perfect.
Cool.
Thank you very much.
Thanks all.
All right. Moving right along. Our next speaker was unable to join us, so we have a recording from Morgan Johnson. Morgan is working on a master's in fisheries and wildlife with Dr. Angie Dewar, and the Holt Award.
Hi, my name is Morgan Johnston. I am a second-year master.
1 second, please. The Holt Award is the Curtis and Isabel Holt Education Award. This provides funds to students doing marine science studies to complete their research, and honors Isabella Holt, who was an OSU alum from the class of 1916. So that's very cool. All right, we ready?
Hi, my name is Morgan Johnston. I am a second-year master's student advised by Dr. Susie Piacenza and Scott Happel of OSU, Leif Rasmussen from ODFW, and Aaron Berger from NOAA. And I'm here to tell you about how I count fish. And no, counting fish isn't quite as simple as Dr. Seuss made it out to be. For my research, I'm estimating the abundance of rockfish along the Oregon coast using some exciting tools like video landers and species distribution models. Before I jump into how these tools help me estimate abundance, let me introduce you to the star of my research, the yellow eye rockfish. These beautiful orange fish are special because they can live up to 150 years old and are one of the largest of the Pacific rockfish species. These fish are also special because they're the last reminder of the West Coast groundfish crisis. Back in 2000, the Pacific Fisheries Management Council shut down all fishing for 9 important rockfish fisheries due to collapsing stocks. Over time, and with rigorous actions by management agencies, such as implementing area closures and reduced harvest levels, these 9 species have rebounded, but yellow eye has yet to cross the rebuilding threshold, supposedly. Anecdotally, local anglers and trawlers say that they see yellow eye all the time and that there are way more out there than the assessment says there are. So why doesn't the stock assessment say so? Are we counting this fish wrong? Ironically, when you close a fishery to let the population rebound, you also lose a lot of important data. If people aren't allowed to catch fish, then we can't count them well using traditional stock assessments. But what about fisheries independent surveys? Well, NOAA conducts groundfish trawl surveys to try and capture what we don't see in active fisheries. However, yellow eye live in rocky reefs where these trawls cannot go. So now we see the problem. The stock assessment doesn't have the data to see the whole picture. This is where my research comes in. I'm using video lander surveys and species distribution models to help us count these elusive fish and to help the stock assessment get a better picture of how many yellow eye are actually out there and where. First, let's talk about video landers, what they are and how they can help us with this problem. These landers are basically video cameras with lights mounted onto a crab pot base and are tossed over the side of a boat to land on a rocky reef and just record what's down there. ODFW has been collecting video lander footage across the Oregon coast since 2009. They were built to be deployed into highly complex and rocky habitat, which happens to be where yellow eye love to live, and take a snapshot of all the other species on the reef. This method is preferred to observe sensitive benthic species like yellow eye because no fish are removed or habitat damaged. Landers are easily deployable, especially in rough Oregon seas, and are more cost-effective than ROVs. The best part is this data gives us a count of how many fish are actually down there, rather than fishing estimates that rely on fish being catchable. And the count data is exactly what I needed to build my species distribution models. Now let's get into how I'm going to use the data. I am building a predictive SDM, which is a statistical tool that combines observation data with environmental measurements to predict distributions of an animal across space and time. So we already know yellow eye prefer rocky reefs and roughly how many might be on a reef, but they don't live on all rocky reefs across Oregon. So what makes those areas special? What other habitat features can help determine how many there are across the landscape? Hopefully, with my model, we can link environmental effects like bottom temperature, chlorophyll A concentrations, and ocean currents to yellow eye abundance in a way that can help us narrow down the possible rocky reefs inhabited by yellow eye. After preliminary model selection, this is what my model will include. Don't worry, we won't go into the weeds on this, but the hope is this model can tell us a location on some day when the ocean conditions were X, Y, and Z, how many fish might be there. And when you add all those numbers up from the resulting maps, you get a relative index of abundance that can be directly incorporated into stock assessment models, hopefully helping to give a better picture of the yellow eye stock status. Now, if I can leave you with one take home today, it's that there are many ways you can count fish, but the trick is finding the right data and the right methods to match the ecology of your species. So with that, thank you all for your time today. I'm sorry I could not be there in person. Thank you to all my advisors and funding sources, and a special thank you to the Hatfield donors for supporting my research. Bye.
Thank you very much, Morgan. And there are a couple folks from ODF&W here, so if you have questions about some of that work, the folks at ODF&W might be able to help you out. So our next presenter is Luke Bobet, who is working on PhD with IB and Dr. Spinagle. Luke received 2 different awards. The first one was a 2003 Boza Award, and that honors Marianne Boza, who was the OSU Hatfield Marine Science Center program manager here from 2010 to 2017. Luke also received the Markham Award in 2020, and the Markham Award is the namesake for this event today. The Markham Award honors Mimi Markham's longstanding support of the Hatfield Marine Science Center. So I'm going to hand it off to Luke.
Can you hear me? Let's see. Is that a yes? Great.
Ready?
Yep, ready. And similar to Morgan, I'm going to talk a little bit about species distribution models, but a different application and their use for understanding phenological and spatial overlap between northern anchovy larvae and their prey. And when we talk about phenology, this refers to the timing of life history events, and these can be influenced by environmental factors for a lot of different species. And one example of a species whose phenology is being altered by the environment is northern anchovy spawning phenology. And we're specifically looking here at the northern subpopulation of northern anchovy from British Columbia down to Cape Mendocino, California. And we know that their spawning phenology has changed because we found larval anchovy off the coast of Newport during the winter in the years during the warm blob marine heatwave event. And you can see those red bars appear during 2015 and 16. And this could be bad news for these anchovy larvae if they're present in the environment when their prey have not yet become abundant in the environment. So this is one possible case of a match-mismatch hypothesis scenario, which essentially says that a consumer's fitness will be maximized when its phenology is synchronous with a resource that it depends on heavily. I was also interested in a spatial component of the match-mismatch hypothesis because resources in the ocean are distributed patchily. So that might be important for understanding larval anchovy fitness. And so my question with this project was whether spatial and temporal overlap between zooplankton production and larval northern anchovy abundance influences the fitness of those larvae. So to answer this question, I'm still in the process of using this 4-step approach that involves a species distribution model for anchovy larval abundance, a model for their recent daily growth, combining those models to create predictions of larval anchovy growth rates and relating those growth rates to adult abundance. So the first step there, the species distribution model component, uses inputs from regional ocean modeling systems, including a lot of physical ocean parameters, as well as from biogeochemical models, critically including zooplankton abundance, to predict the abundance of anchovy larvae measured by our lab using our ichthyoplankton imaging system, but also by NOAA using bongo net trawls over a longer time scale. And so the result of this looks similar to this, where you can map the abundance of anchovy larvae across the landscape, and hopefully this reflects changes in their phenology as a result of changing environmental conditions. So the second portion there is a model of recent anchovy growth, and that uses the same inputs as the species distribution model, but then predicts the growth of anchovy larvae measured from their otoliths in those growth rings that you saw. And growth rates are an important measure of fitness of larval fishes because these guys are small, they're very vulnerable to predation and starvation. So the faster they grow, the faster they move up this food chain and avoid those pressures. So then the third step combines that species distribution model and uses inputs that I've talked about already, critically including zooplankton abundance, to make estimates of larval anchovy growth And then I'll compare those growth rate estimates to observed growth rates that were withheld from the original training data set used to construct the growth model. And if those observations are similar to the estimates, hopefully I'll be able to hindcast larval anchovy growth with some degree of accuracy and aggregate those hindcasts within each year and compare those yearly average growth rates to annual anchovy abundance, which you can see is pretty variable for the northern stock of northern anchovy. So being able to understand what drives some of this variability would be very valuable to the management not only of northern anchovies, but of the different species that depend on them, like seabirds, other fishes, and marine mammals. So I'm very grateful for receiving the Markham Award that helped me get going on this project, and also for the other members of my lab who have helped collect data for this project, and for our collaborators at NOAA who have lent some data to make this possible. And thank you all for your attention.
Thank you, Luke. Any questions for Luke before we let him off the hook? All right, thanks, Luke. Our next presenter also got a Markham Award, but in 2021, Maya Fazell is working on a PhD in IB with Dr. Bruce Mengi.
Right.
All right, you ready?
Can we all hear me okay? All right. I'm ready. All right. Yeah. Hi, everyone. My name is Maya Fazell. I'm a PhD student in the Lubchenco-Mengi lab in the Department of Integrative Biology here at OSU. And I'm going to be talking to you a little bit about how climate change might be impacting a relationship between some snails and some algae out here on the coast. So what is the intertidal? It's this area between the land and sea where waves are regularly covering the rocks, but it's never fully covered by water and it's never fully out in the air, right? It's this transition zone between these two areas. And what that means is that it's a really stressful environment. These organisms are seeing huge temperature changes, huge changes in the amount of water and humidity that they're exposed to, and that leads to it being a really dynamic and really interesting place to study. It also is made up of 3 major zones. So you can see here from left to right, there's the high, mid, and low zone that are all really distinct from one another based on how often the waves are crashing, dictates what kind of organisms can actually live there. And so I primarily study the high zone, and as you can expect, that means that it's the highest point that still is regularly covered by waves on a daily basis. It's mostly made up of algae and barnacles, and they're really resistant to drying out in order to be able to survive in this environment. So here's some of that algae here that I study. It's what we call turfy algae. So it's pretty big, but not as big as what you might see in the low zone or below the water. It's got these beautiful fronds, and it's a really good environment for small creatures like this. This is a limpet. I think they're so cute. They're little snail herbivores that live in this algae up in the high zone. And what's really interesting is that we don't really know what the relationship is between these two groups. So you might be thinking, one's an herbivore and one's an algae. They surely have a bad relationship, right? But we don't really know actually what that looks like, whether the limpets are feeding on the algae or what's going on. And most importantly, we don't really know how that relationship is going to be changed by climate change, especially as temperatures start to increase. We start seeing a lot more of these heat waves that we've been seeing out on the coast. How is that going to be altering how these two groups interact with one another? So how do we find this out? This is my experiment that I set up last year here at Hatfield. It's a bunch of tanks with a bunch of limpets that I was exposing to different temperature regimes and different types of algae to see what they're actually feeding on and what may be going on out on the rocks. So what's happening now? It looks like those limpets don't really feed on this big algae. Not only is it just way bigger than them, these limpets are about the size of one of your fingernails, but it's also pretty tough. So it's hard for them to get through. They instead mostly feed on what we call microalgae. So if you've been out tide pooling, it's kind of that general sludge that's all over everything. They love to eat that stuff. It's much easier for them to get to. But what might be happening as time goes on as temperatures increase? Algae go through this process called bleaching when they get really stressed out, where they start to get less tough and they aren't able to produce a lot of their chemical defenses that they're able to usually, which makes them a lot easier to feed on for little organisms like the limpets. So as these temperatures are increasing, we start to see that the algae is more susceptible to predation, basically. So what does this mean for this community? It means that we could be seeing smaller algae populations as time goes on, especially as these hot and dry conditions mean that that microalgae, that sludge, may not be as able to grow as things get hot and dry, right? And this could lead to some really long-term changes out on the coast. Like I said, this high zone is mostly algae and barnacles. And so if you see a big reduction in one of those, the community is going to look pretty different in the next 10, 20, 30 years, right? And we have a precedence for this in the other intertidal zones as well. So the low and mid zone, we've been seeing some pretty drastic changes to what the community looks like. And it doesn't really seem to be bouncing back to what it originally was, which is a little bit concerning, right? It's kind of permanently shifting over. And why does this matter? Obviously, if you're here, you care about the coast, and this could be a pretty permanent change, which is kind of concerning, right? And it also serves as a litmus test for other ecological communities. Like I said, this is a pretty stressful environment. So we kind of start to see a lot of those changes here now before they come to other communities, right? Wow, look at that, I finished early. And with that, I want to thank some of my funding sources and my lab group, and thank you all for listening.
Nice job. Any questions for Maya? You have a very, oh, there's one right there.
Hey, I had a question. So just to confirm that the limpets, they're actually eating like phytoplankton then that sticks on the rocks, or that microalgae. What are those populations like?
Yeah. So it can be phytoplankton. It's just a lot of small microorganisms, right? So a lot of it is phytoplankton or other just sorts of tiny creatures that are growing out on the rocks. And they kind of come in and either get washed up or they sort of grow over time in like little pools and stuff. It's really quite gross. But yeah, yes.
I think that's what he studies, by the way, just to say that out loud. All right. Thank you very much, Maya. Appreciate it. Okay, our next presenter is Spencer Luna, who is a PhD student in the comparative health sciences with Dr. Schubinger. And Spencer received a 2022 Markham Award.
Okay, so hi everybody, thanks for being here. So I'm Spencer. I actually have changed departments since I got this award, so I'm now in the Department of Microbiology, but the research is still the same, so we'll keep going. So I will be talking today about my work on deprivation and microbiome studies in Pacific oysters. So Pacific oysters here on the West Coast, this is a pretty big industry. It's about $100 million a year in production of shellfish, about a third of the total for the US. But these oysters can harbor Vibrio perihemolyticus as well as other Vibrio species. So Vibrio is an abundant genus of marine bacteria. They're found all over the world. Several species can be pathogenic in humans, and also we see several species that are pathogenic in both shellfish and fish, in particular with oysters. So, so the pathogens of Pacific oysters that I work with are in particular Vibrio coriolyticus. So this is a pathogen that kills larvae in hatcheries where they're producing oysters for distribution to farm sites. It can also kill spat, which are just newly metamorphosed oysters. So then also I work with Vibrio perihemolyticus, which is a cause of foodborne illness. It's often contracted from eating raw shellfish, and it's destroyed by cooking, which is good if you like to eat cooked oysters, but not so much if you're trying to eat oysters raw, which I like to do. So for that purpose, what you can do is you can use post-harvest processing. So after collecting your oysters, you could treat them using either freezing or high hydrostatic pressure or deprivation. So high hydrostatic pressure and freezing kill the oysters that can change some of the qualities that people like in a raw oyster. Deprivation, on the other hand, this is the process of holding shellfish in clean seawater and just allowing them to purge over time. This has been used for a long time for reducing pollution-associated bacteria like E. coli, things like that, but work on Vibrio has been a little bit a little slower in getting there. So just to briefly talk about the system I work with, so this is a deprivation system. It's basically a recirculating seawater tank. It has filters built into it. It has a temperature regulator, so you can set it to a certain temperature. This is to avoid having a temperature that's too warm, and we'll be encouraging the growth of this bacteria because we're trying to keep that Vibrio from growing within the system. And then also we have a UV sterilizer built in just to kill any bacteria that are released into the seawater. And then we have holding chambers where the oysters are kept. And so you can see in this picture, this is what the oysters look like when they're actually in that chamber. And so the way this works was 24 hours before we wanted to actually set up a deparation trial, we would fill the system with seawater and allow it to run at the desired temperature just to ensure that the seawater we were using was sterile. And then we distributed our oysters into the holding chambers. So every day after that, we would collect oysters, shuck them, homogenize those, and then plate on selective media where only Vibrio will grow. And that allows us to see how many Vibrio are present in these oysters over time, over the course of deparation. And what we end up seeing is that, yeah, using deparation, you can significantly reduce Vibrio parahemolyticus in oysters. So the Pacific oyster, the one I'm talking about today, is on the left there. And just, yeah, in the first 1 to 2 days, you'll see a reduction, a pretty dramatic reduction, really, several fold. And then from that point on, it kind of slows down in reduction. So I know that it works for, or that deparation reduces Vibrio, but I wanted to see, does it also affect the overall microbiome, which for mycorrhizis is just the diversity of bacteria present in this organism. So does deparation affect that? Maybe also selective breeding for disease resistance, which is something else I work on. So yeah, just with regards to that, like, do we see particular bacteria that are associated with disease resistance? And also, does this deparation impact certain bacteria more than others? Do Vibrio persist, or are they also reduced like parahemolyticus is? So just the process for that, that I've been using is I collect tissue samples from oysters. So these can be different tissue types from the gut, the gills, things like that. And then also looking at oysters that are bred for disease resistance versus oysters that are not, and seeing, do we see a difference in the microbes within these different lines of oysters? So just the brief process there is I'm extracting this, specifically the microbial DNA from these sampled oysters, extracting that. And I specifically want to look at just the microbial DNA, so ignoring any oyster DNA that can kind of contaminate or change the results. So then I'm using polymerase chain reaction, or PCR, just to amplify that extracted microbial DNA, focusing on short sequences that can be used to identify all of the different types of bacteria that are present in an oyster. So once I've done that, I'm now at my kind of last phase at the moment, where we're waiting on my DNA sequencing results to get back. So for that, it was just, I was confirming that PCR worked. I had amplified DNA to work with that would show me what oyster or what bacteria are present in these different oysters. And then I'm now getting that ready. It seems like it's worked, and I'm getting ready to submit for sequencing. So I'm hoping that by the late summer, I should have that DNA sequence data back, and I can see, do I see any treatment effects? Do I see that deparation has affected the microbial diversity or abundance, as well as looking at those two different disease resistance lines to see, do I see a difference there? So yeah, with that, I'd just like to thank everybody for being here, and thank the maybe Markham Research Board for funding this work. Thanks. You can send me an email if you need to.
All right, any questions for Spencer? Okay, off the hook. All right, our next presenter is Matt Vaughn, who's working on his PhD in IB with Dr. Hinkle, who also received a 2022 Markham Award.
Yep. All right. So did you guys know that shrimp burrow? Because I didn't know that until I came here for my PhD. So my research is looking at ghost shrimp, and ghost shrimp, they burrow down into the sediment on the sea floor. Over time, they continue to construct their burrows, and these burrows get really deep. The ghost shrimp spend the majority, if not all, of their time in their burrows. And when they're constructing their burrows, they bioturbate, which is shown here. It's basically just kicking up sediment, and this can impact filter feeders. They also destabilize the sea floor, and this can impact other burrowing organisms and invertebrates. Now, ghost shrimp, they also alter the biogeochemistry. So when they burrow down, they actually oxygenate deeper sediment. You can see that here, the lighter sections are oxygenated, and they bring inorganic solutes up to the sea floor. So this can have positive or negative impacts depending on what the other invertebrate species is. And ghost shrimp are common in the estuary. In Yaquina Bay, we have 2 species. The ones in pink are more abundant, and the beige one is larger and less abundant. So in 2019, we actually found a population offshore, really large population. We've sampled this area a ton, and we haven't found a population this dense, leading us to believe that it's a new population. So you can think of these offshore shrimp more like crawdads. They're really big compared to everything else out there. And most of the knowledge that we have on burrow impacts comes from estuary research. So my project is looking to compare the structure and behavior between these two populations. So to do that, I collected shrimp from the estuary at low tide using a shrimp gun, basically sloping them out of their burrows. And I collected offshore shrimp using a box core, which is basically like a giant claw machine that penetrates into the sea floor. Once I collected these shrimp, I put them in these large tanks and allowed them to burrow for 2 weeks. I then applied an epoxy resin, which basically created these hardened resin casts of their burrows. So I could excavate these casts, and then I could take a number of different measurements, such as depth, total length, volume, as well as measures of complexity, like how many turning nodes or sections there are, and how many shrimp are in the actual burrows as well. But just like building a house, here we go, the structure depends on the behavior of those involved. So behavior can tell us which shrimp are building what burrows, where and when, and why behavior might matter, and how it could result in different burrows. So the pink lines here, they're the estuary shrimp, and the teal are the offshore shrimp. The main takeaway message is that the number of burrow openings change over time. So this could indicate that the behavior could also change. So to look at behavior, I set up shrimp in these large ant farm tanks and filmed them for 2 weeks. And I paired this with in-person observations. From this, I'm able to figure out how much time they're spending doing what behaviors and what sections of the burrows they're hanging out at. And I can also estimate how much sediment they're compacting versus excavating, and also how much sediment they're oxygenating. Again, this is important for different invertebrates they live near. So looking at some of my results so far, the estuary shrimp in pink, they burrow deeper than the offshore shrimp, and they also burrow at a faster rate. I think these behaviors are due to the different environments that they're from. So estuary shrimp, they have deeper, longer, and more complex burrows. They also excavated nearly twice the amount of sediment as offshore shrimp. And looking at their behavior, the estuary shrimp, they initially spend a lot more time burrowing compared to the offshore shrimp. After the first day, this levels out, but the estuary shrimp, they then spend more time compacting and ventilating compared to the offshore shrimp, which spend more time grooming. And yeah, where are they hanging out in their burrows? So the estuary shrimp, they never left their burrows. They spent most of their time at the bottom, and the offshore shrimp, they spent more time at the top sections of their burrows, and they also occasionally left their burrows. Again, I think this is reflective of the different environments. So to wrap it up, I found that burrows and behavior of offshore shrimp are different. Further research can be done to better understand the effects offshore shrimp might have on the surrounding offshore community. And with that, I would like to thank you for the dollars to conduct this research and for everyone that supported this project.
Okay, I have a question. How many hours did you spend in the little dark room watching the little shrimp burrow?
Well, I recorded them for 1 hour initially, and then 2 lots of 20 minutes each day over 16 days. So do the math, it's probably about like 100 hours in that dark room. Yeah, my eyes have changed now. They've evolved.
Got more vampires coming through.
Yeah.
That's great. Any other questions? I didn't mean to steal that from everybody. All right. Thank you very much, Matt. Okay. All right. Our next presenter, Cheyenne Jarman, is working on a PhD in IB with Dr. Novak. Cheyenne, has received a 2022 McNeil Award. The McNeil Award is kind of a special award that we have access to that allows us to fund projects that don't fall into the typical box of marine research in the standard way. And you're going to find out why in just a second. All right, take it away.
Hear me okay? Cool.
Ready?
Yeah. So kelp forests are beautiful and complex ecosystems that provide food, habitat, and shelter for life both below and above the sea. My research career thus far has just investigated a small fraction of the myriad of interactions and dynamics that occur in this ecosystem. If we look at just kelp alone, we see that kelp can exhibit two different dynamics, one where it persists over time with highs and lows, and others where it's completely absent for long periods of time. Yeah, so my dissertation works specifically focuses on this persistent kelp population and uses a combination of empirical ecology, theoretical ecology, and machine learning to build mathematical models that explain these dynamics. Now, this work is inherently interdisciplinary, and while I was prepared for some of the challenges from my background in marine biology and computer science, there were areas I definitely needed to strengthen, which led me to the Santa Fe Institute's Complex Systems Summer School. So the Santa Fe Institute Complex Systems Summer School, I was able to go to this because of the McNeil Award. It is a 1-month-long program that runs at the Santa Fe Institute in New Mexico. And Santa Fe Institute is a nonprofit research and education center that has a mixture of internal and external faculty from all around the world that study complex systems, which they define as a system in which multiple agents are interacting with one another and their environments, which can give rise to emergent properties, which includes ecosystems, cities, the internet, and such. The Complex Systems Summer School provides a wonderful opportunity for individuals like myself to learn from and alongside other ecologists, social scientists, physicists, economists, and so many more subject matter experts. In my cohort alone, there was quite the range of representation of the different disciplines, which you can see in this pie chart here. And the summer school is unique in the fact that it's not only lecture-based, but it's actually requires students to provide or use their previous and freshly learned knowledge on projects that we work on throughout the month. And so for that month, I actually worked on 2 projects. One was the complexity of music, and the other one was looking at the complexity of human interactions in community engagement. And for the sake of time, I'll only be talking about this community engagement project. So after the first week of lectures, students were able to pitch ideas of projects they wanted to work on. And a cohort member of mine threw up this graph on the board saying, is there this kind of ideal level of conflict that societies can have that keeps as many individuals engaged as possible, keeps everyone talking? And as an ecologist, my mind immediately went to the intermediate disturbance hypothesis that shows if you have this disturbance frequency on the X and species diversity on the Y, it's going to be highest at this middle point of intermediate levels. So naturally, I teamed up with her along with many others and took a network approach to study this. So we first defined 2 different nodes or individuals. We gave them opinion values, which could be -1, 0, or 1, and then tolerance values, which were 0 of you're completely intolerant of another person's opinion if it disagrees, or a tolerance value of 1 where even if you disagree, you'll still maintain this edge connection. And then we then calculated what that edge connection would be through 2 different terms. One was a disagreement value, and then the other one was a tolerance value. So for our 2 little nodes here, they disagree because one has a -1 opinion, one has a +1, and the blue dot is intolerant, so they're no longer going to maintain that edge connection once we kind of shock the system. So we then expanded this out to larger networks. We used 2 different types. We used a random network where you have a bunch of different nodes, they're all connected in different ways, or a stochastic block model, which has 2 kind of main points to your networks, but they're still maintaining edges between them. We then kind of generated this with a uniform distribution of opinion and tolerance values, and then we shocked the system to see if nodes would maintain their edges once they kind of realize that the other person maybe thinks a different way or has different tolerance values from that. And our preliminary results, if we look at how engaged are people who disagree, which is our weighted edge, we do kind of see this hump-shaped curve. And so we have opinion divergence on the X and that weighted engagement on the Y. And the random is in blue, stochastic is in yellow. And we do see that hump-shaped curve. And these are very preliminary results, I should state. But this work is still ongoing, which is really amazing. And it does look like that Connell graph from 1978, which was really cool. And so that leads me to some of my takeaways from this. Definitely embracing the challenge of interdisciplinary work and approaching it with genuine curiosity. I think the Santa Fe Institute does that really well. All the faculty and students were really interested in what other people were doing and really interested on how they can collaborate with one another in that way. The second question is asking a simple question can open up a lot of doors. My friend, when she threw that graph up on the board, had no idea what she was getting into. And finally, thank you to the McNeil Award for affording me this wonderful opportunity that's not only expanded my research portfolio, but has definitely made me a stronger collaborator. Thank you.
Thank you very much. Any questions? All right, off the hook. Okay, so our next presenter is Jennifer Waldo, who just defended on Tuesday. Congratulations. Jennifer has completed her master's degree. I'm assuming you completed and it was positive, and yeah, I haven't heard. I should ask that before. In fisheries and wildlife, or fisheries and wildlife with Dr. Jones. Jennifer received a 2023 ODF&W award. And the ODF&W awards are awards that help students work with ecological processes that shape nearshore marine communities in Oregon waters. So I'm gonna let Jennifer take it away.
Okay, I think this is on. All right, ready? Hi, everyone. Thank you for welcoming me here today. As Cinnamon mentioned, I'm a recently defended fishery science student. Today, I'll be presenting my work that's funded by the Oregon Marine Reserves. So humans are deeply connected to the oceans. Oceans provide us with food for sustenance, hold personal and cultural significance, and are overall valued from not only a tangible standpoint, but from an aesthetic one as well. Think about the beauty of the Oregon coast. The oceans, and thus our own livelihoods, though, are in peril. Marine ecosystems and the people who depend on them are facing the impacts of climate change, including here in Oregon. And whether realized or not, human behavior and action partially contributes to the degradation of our marine environments, either directly through personal interaction with the ocean and coast, or indirectly through resource use and lifestyle choices. But at the same time as our actions and behaviors can contribute to ocean change, they can also powerfully contribute to a sustainable ocean future. But these actions are often misunderstood or misoverlooked. So how can we motivate people to take action in response to ocean change? How can we motivate people to engage in these powerful, influential actions? Which brings me to my two overarching research questions of my master's. It was first, what are the motivations behind taking climate action specifically in response to ocean change, a threat that is often perceived as distant? And how can we use psychologically wise, evidence-informed communication to do so? So I looked first at people's knowledge of approximate threat of ocean change right here in Oregon, ocean acidification, and how we can increase knowledge to see if that engages action. Second, I looked at how we can connect people further to these issues through a concept I'm calling connectedness to the coast. This is the awe you get when you think about the ocean and you stare out at its beauty. Third, I looked at social norms of taking climate action. So if my friends and family are taking climate action, and am I more likely to engage in those actions, especially if they approve of me taking those actions? And finally, I looked at increasing people's efficacy beliefs that they can take action. This is the confidence that you have that you can engage in an action and that that action will have an influence or impact. And so there's these two broad buckets of actions or of motivations. And I took a two-wave survey approach. The first was an online survey of Oregon residents that received 1,414 responses. And this was specifically to look at people's motivations to take action. I then took these motivations and put them to the test through a messaging experiment of visitors out on the Oregon coast, which received 2,414 responses. And so through my online survey, I found that all of the motivations I looked at were significant, but some key things I want to highlight from this is that efficacy beliefs were the highest motivator for taking climate action. Comparatively, ocean change beliefs were much lower. But do these results hold true when we put them into actual communication application, which is what my experimental survey intended to do? Through my experimental survey, I found that only a message that targeted people's efficacy beliefs, so their confidence in taking climate action, motivated them to intend to take those actions. And so I highlight 3 questions we should ask ourselves when communicating about these threats. One, what about ocean change is going to be prevalent and important to our audience? I saw that ocean acidification didn't seem very salient to our audience. So what would be? Second, if my goal action is something that's social, if I was looking at people's intentions to make climate action a social norm, then maybe I should look at more social motivators that we saw earlier. And finally, efficacy beliefs were found to be a promising target in only a 30 to 60-second interaction I had with folks. So how much time do we have available, and what's going to be most powerful to use to motivate them? All right, so that was a very brief overview of my master's. I just gave a 45-minute presentation the other day, so I'm happy to dive deeper if anyone's interested. Thank you so much.
All right, any questions for Jennifer? 1 second, please.
Okay, so it looks like efficacy, right, seems to be the kind of bigger driver. I guess, how do we inspire that in people when we're talking to them? Often I hear, like, you know, the ocean's so big, these problems are so huge, like, what can I do? What can we do when talking to other people, you know, stakeholders or whatnot, to kind of inspire this efficacy and make it, you know, not seem so hopeless?
Great, thank you so much for that question. It's a great one. So the question was, how can we inspire efficacy so that people take action? In my research, I was mostly interested in these more social actions of encouraging your friends and family to take action with you or talking to others. And so I think that personally, I think focusing on these social actions that can be somewhat easy for people to take, can be more influential, particularly with climate change, which is a systemic issue. Focusing on social actions has evidence that social mobilization efforts can lead to powerful political, systemic, structural changes that thereby lead to people's ability to take other actions like bike commuting and things like that. So I think focusing on really tangible actions people can take within their daily lives helps to increase their efficacy to take those actions. Thank you.
Thank you very much. We are almost there, everyone. We have one more presenter. Jake Marshall is working on his master's with MRM, and Dr. Hunsinger received a 2023 WIC award. And the WIC award honors Oregon's very first Sea Grant director, Bill Wick. So I'm going to hand it off.
Cinnamon. Yes. Awesome. Hi, everybody. My name is Jake Marshall, and my presentation is titled, Where the Hake Even Are They? Spatial Distribution Modeling of Pacific Hake. Here are some of my collaborators and funding sources, thanks to the Bill Wick Award. Quick overview for my quick talk. I'm going to be talking about Pacific hake, some pressures that the hake fishery are encountering and will encounter in the future, the spatial modeling work that I've been doing over the past year and a half or so, and the use of these models. So Pacific hake, also called Pacific whiting colloquially, is a semi-pelagic ground fish found on the North American West Coast. It makes up a large commercial fishery and is mainly used in the production of surimi or imitation crab. Pacific hake is the largest commercial fishery on the U.S. West Coast by abundance. It has an annual yearly catch of around 250 million pounds of fish, supports around $350 million of U.S. economic impact and 4,500 jobs in the U.S. It is an annually migratory species. It spawns off the coast of Southern California and Baja in the wintertime. It then travels north to the coast off of California through British Columbia to feed in the summer. During this northern migration, it's commercially fished by the US and Canada because it's in both of those national waters, and it's therefore a transboundary species, and it is co-managed by the two countries. Importantly, it's been seen recently that temperatures had impacts on where hake are distributed along the coast. We see that warmer years have had, impacts on where we can find hake. This is very important because of stressors like climate change. Things like marine heat waves have been impacting our coasts over the past few years. There have been many ecosystem effects like changing distributions, a lot of die-offs, a lot of crazy things have happened. Fish distribution shifts have been one of these things. And importantly, marine heat waves are seeing an increase in both intensity and occurrence, and are predicted to increase in both of those through the end of the century. So we need to find ways to mitigate the impacts that we are expecting to see. Some other pressures in the Pacific hake fishery are geographical boundaries. The one that I'm focusing on in my thesis is the US-Canada border. Essentially, the US fishes in US waters, Canada fishes in Canada waters. Here we see 2 years of hake presence. In the 2023, we can see that there were not a lot of hake present in Canada, which led to their worst fishing year in about 20 years. So that brings us to the current situation that we have, which is basically we have the hake fishery that is being both affected by climate change distribution shifts, so it's affecting where we can find hake alone. And we also have geographical restrictions of fishing, like the international border. And other geographical restrictions are things like offshore wind energy, where we won't be able to fish for hake. And my project is based around the idea that spatial modeling can help guide management and fisheries operations decisions in these complex and uncertain situations. So a little bit about the modeling work that I've been doing. I've been taking the hake survey data, which basically says like where the hake are, how many there are, linking them spatially with environmental data like ocean temperature is my main driver. And we find this relationship that is essentially the model. Once we have that biomass model for the hake, we can then give it future predictions of temperature that we can infer where the hake will be under those temperatures. And the rightmost map shows us the actual 2024 prediction that I have so far. And this wouldn't be possible without the ocean temperature forecast that can be made up to 8 months in advance. So we can actually have hake forecasts up to 8 months in the future. Once we get these distribution outputs, these forecasts, we can then divide them among different regions that we care about, like north and south of the US border, inside, outside of offshore wind energy areas that can then be used to help fisheries management and fisheries operations decisions. Fisheries management could use these outputs to, there's a little fish manager. They can use these to set catch allotments, and fleets can also use these to make decisions, because a lot of the hake fishery also fishes for pollock up in Alaska. If they're expecting a pretty bad fishing season around Canada or the US, they can just make the alternative decision to, instead of not catching hake here, go not catch hake and catch pollock somewhere else. So thank you everybody for your time listening. Thanks to my advisor, Mary, and other people who've helped me on the hake survey and my trusty computer and all of its analysis. Thank you.
All right. Any questions for Jake? All right. Thank you very much.
Thanks.
All right, everyone. We have done it. Can you give everybody one more huge round of applause for all their effort? Everyone in this room knows that our graduate students and our undergraduates are just key to accomplishing our mission here at Hatfield. And so I am so proud of everybody that presented either a poster or the work that they're doing. I often give tours. You all see me out there, and I talk about the diversity of the work that happens here. Did you see it? I mean, like, we talk about all sorts of amazing things here. So again, thank you so very much to our students. I really appreciate it. And thank you all for being here and being a part of our students' success. Again, thank you so much. I also want to just say that this event does not happen by one person alone. There is a whole team of folks that help take care of moving the furniture and providing food. And so the operations team team of Tammy and Miguel, the director's office team of Shannon and Katie, the folks that helped the room today. So we had Taylor, we had Riley, we had Marnie, all here making sure that this all went well. So thank you all for your help in making this event work. I appreciate you as well. All right. Before we switch to our very next thing, we have one more little announcement to make, and I'm going to hand the mic to Matt.
Yeah, so after dinner, some grad students are going to head to Beer One, grab some drinks. Would like to invite everyone in the room to continue the conversation. So feel free to come to that. We'll say 7 p.m., but we'll probably trickle in whenever dinner ends.
All right. Thank you very much. Okay. With that prelude, there will be a dinner that will be served up on the atrium of the second floor. For those folks that have been invited, our award winners, their mentors, and our interns that have joined us for the summer, you're welcome to join us for dinner. And for those folks that might know folks that have posters, just remember that part too. We've got some posters for folks to pick up. And again, thank you so much for being here and being a part of this event. So thank you very much. Dinner will be served in about 15 minutes upstairs. So you got a little break. Oh, how many people? We got a photography request. My Night Talk folks, you are now famous. You need to get into a photo. Can you come up front before you go upstairs? Thank you again, everybody. Thanks, everyone online. We're going to end now.